Hello, everyone. My name is Bogdan Budnik. I would like to start our first session on the presentation of initiative from the Biology of Disease initiative at HUPO. Today, we have the first two initiatives is going to represent themselves. And it is my pleasure to introduce Tinan Goa. He from Westlake University. And today he will present AI initiative that I'm very much excited about to share with everyone, because in my opinion, this initiative would work with all other initiatives that are in our Biology of Disease group. And that's why it will be very good that he would represent what the view of the AI in proteomics and how they plan to work with the data in near future. Okay, without further ado, Tinan, stage is yours. I would like to add that all the questions during the talk, please put in the chat, and I will read them in the end, and Tinan will have a time to respond to those in the end of his talk.
Yeah. Hey, thank you. Can you see my slides and hear me? Yeah, it's a great pleasure to present the HUPO AI Proteomics Initiative and the recent progresses. As some of you might know that we started this initiative during the Dresden meeting last year. Today, my talk will contain several components. So the first is to briefly introduce the history of this very, very short initiative. And second, I will introduce our recent discussion, and then which has been condensed into a paper, perspective paper, to prioritize the strategical priorities for AI proteomics. And the third is to one of the actionable points that we have been discussed and actually executed is to introduce the Proteome Big Data initiative or data resource we call the MassNet. And then we are starting another concrete action, which is AI competition. And third, another very exciting field, which is AI virtual cell. I will introduce our recent progresses and plans. So I've been thinking about what name should this call. Some people say AI and proteomics. Some people say AI for proteomics. So when I talk to computer scientists, so they will say AI for proteomics because they are AI, they are for us. But for proteomics people, normally we see computational proteomics. So I'm struggling what's the name, and eventually maybe just to say AI proteomics. They are equal and they are supporting each other. So last year in Dresden, thanks to the support from Bogdan and many others, then I actually gathered about 20 to 30 scientists from different countries. And so they all support this AI proteomics initiative. Some of them physically joined the Dresden meeting, and we had a very good discussion, heated discussion in Bioinformatics Hub. And later, some of this discussion has been condensed into paper. So this year, after the Dresden meeting, we have some online discussion, several online discussion, a lot of discussion in Google Doc when we try to condense the ideas. So eventually we get 62 scientists interested in AI proteomics. And these are the names. And we have, I'm in Asia, and we have Professor Bin Zhang from Baylor Institute, Baylor Medical College in the US, and Matthias Mann from the Europe. And we also have quite a few people from the proteomics community. You can see their names from Pride and many other institutes, also from computer science. So some of these names you may not be very familiar because they are from a different field. And there are also people from industry, from pharma company, from AI companies, in the leading AI companies in China. And in future, we hope to have more people from other countries, from AI, from the other field join us. So we have published a perspective in arXiv. This is the first time I published a paper in arXiv, because normally we publish in bioarchive or meta-archive. And this paper is currently under review in Nature Methods. So basically, this paper we try to discuss and present what are the priorities for AI proteomics. In the following a few minutes, I just briefly introduce what we have discussed. So we think we should not only focus on technology, but we should actually aim at solving some biological problem to have higher impact. So we start with the molecular biology. We have a lot of information, but very highly complex and very challenging to predict even a single cell's behavior. So proteins will be important, as all of you know, and we have recent progress in mass spec-based proteomics. We can measure not only protein levels, but also many other aspects of protein and protein network. And we have the very good advances in sensitivity and throughput. So, we have seen a lot AI, successful application of AI in mass spec-based proteomics, but still there are challenges. So the challenges, the very ambitious biological goals, still very difficult to achieve, and we have millions of mass spec files generated by the community in HUPO, outside HUPO, and they're deposited, but only a very tiny fraction has been used for AI training by the computer scientists. And we lack an AI-friendly ecosystem for proteomics data generation, sharing, and analysis. So this is a topic the Bioinformatics Hub and many computational proteomics meetings has been discussed for years, probably for decades. And we lack clear research goals for computer scientists. I will mention later. So then we concluded probably the most burning issue to solve as this initiative is to construct an AI-friendly ecosystem for proteomic data. So we outline the procedure from sample, biological clinical sample, through sample preparation, LC, mass spec, of course, data independent mass spec like Olink can also be included. So to data sharing, so here we highlight is very, very important to not only share the raw data, but also share data in a tensor format, so which can be directly read by computer scientists. And the annotation, so Pride team has emphasized the annotation for this data. And then public, the code, the data should be shared to the entire community and for data analysis. So we're outlining how to build an AI-friendly ecosystem. But of course, this needs cooperation from everyone in the community. So then we prioritized 6 key areas. Most efforts currently are focused on identify, quantify of peptides and proteins. I'm not going to talk about the details. And the next field is a protein complex, PPI, and other molecular interactions. And then spatial proteomics, as you know, they're combining protein expression and spatial images. And perturbation proteomics, this describes the dynamics. I will have more details later. So then multi-omics integration and AI virtual cell. So the number 4 and number 6, I will talk about later on for more details. So if you're interested, you can read and comment in the archive paper. Due to the time limit, I'm not going to talk too much detail about this. So then one of the concrete action this AI initiative is working on is to build a mass net. Probably I don't need to introduce the image net to you. Everybody knows every image in the real world, every object in the real world has an image, so that we can develop a very intelligent AI program. So in the world of mass spectrometry-based proteomics, I think we need some sort of mass net. So it's for every peptide, every protein, we should have an annotated entry. So in the past, when we analyze, or for most of the mass spec software, you have multiple data, and you can sometimes you train a model, a statistical model or machine learning model, for different file, you have different models. For a small batch of file, you have a integrated model. But if we can integrate all this annotated raw data, we can build using pre-training algorithm to build a large AI model. So this can be very different and much more powerful than individual small models. So to capture the granularity and complexity of real proteomic data, more sophisticated models are needed. So we propose that by joining forces, I believe everyone, everyone of you, if you run mass spec lab, probably you have maybe 50% of your data has been published and deposited in Pride, and a lot of data actually not published or on the way to be published. So this data actually can be utilized to build a mass net as a community effort. So we have a perspective under review. So to do something to initiate this MassNet, so MassNet can contain DDA, containing DI, can contain targeted proteomics. So we started with a DDA. What we start is to get all the data from my lab, which is called Gormix. We have a 5.6 terabyte of only DDA files generated by mass spec at high quality ones, which are higher than QEHF, or including triple tops and other higher end mass spec. And we download 3,600 DDA files from iProx and a lot of files from Pride in collaboration with Pride team. So now, after data analysis with Frag Pipe and Sage, we obtained over 1.5 billion high quality mass spectra. And this will be shared to the community very soon. So in our goal, we will build a version 1, which is about 1 billion, and maybe we can build a 2 to 5 billion with a contribution from the community. So next phase is to find who are interested to contribute to this initiative. So next one is the AI competition. You all know how AlphaFold and Rosetta were developed and succeed. So because there's a cusp competition. So then we are talking with a lot of computer scientists, why not have a competition for proteomics? Then we got a concrete plan to submit a dataset proposal to NeuroIPS, which is a leading AI meeting in San Diego this December. And we also get some sponsorship as a AI competition for DDA rescoring. So we will start next month. Also, we are organizing another Zoom meeting with interested colleagues. So next few minutes, I'm going to talk about the very, very exciting emerging field of AI virtual cell. And this is another major initiative starting in this AI proteomics initiative. I think we can have collaborators from many fields, not only proteomics, but also other fields. So since a couple of years ago, I've been very interested in building the digital cells, combining AI and perturbation proteomics. We published a perspective paper to introduce the idea of AI-empowered perturbation proteomics for building digital cell. We call it digital cell. So it will be possible to perturb cells or tissue or animal model in human being with different biological, chemical, physical perturbations, or whatever you call perturbation, and then measure proteome. Of course, one can also measure other omics and the phenotypes, and then we can build a model. So perturbation has been done for a long time, since about 15 years ago when I was a PhD student, but we have a very limited number of perturbations. But then if we have the ability to perturb and measure proteome for thousands of conditions, then this will change. Then AI can be used to model and then predict, and then we can actually, based on the prediction, we can feedback and generate more perturbation. So when we wrote this perspective, this is what we thought about at that time. Then last year, after the Dresden meeting, I organized another Westlake Symposium for the Future of Proteomics meeting in our university. And then 1 week later, we organized a second Pi-Hub Council meeting. So there I proposed, after a lot of discussion with Rudy and people mentioned here, Marcus, Matthias, Chris, and Weilan and Hang. And so they are leading scientists in AI scientists in China. So we proposed the iEast project. Because East, usually I work on human, but human is complicated. So we decided to develop the methodology to building a digital cell using East, because East has a rich data set of genomics and lots of thousands of mutants, and only have about 6,000 genes, and a lot of modelings using East, a lot of images, and like a GFP fusion proteins. So there are databases, there are also AI models or mathematical models, but they don't have a lot of proteomic data for perturbed East. Although Marcus and Matthias, they have both published very nice paper for East proteome describing the intactome and the metabolomics and proteomics, but there's no advanced AI model. So we want to work on this, but not much concrete action. So during the meeting, I saw a interview from Demis Hassabis, and he said DeepMind probably will work on create a virtual cell during the next 10 years. In this very short interview, he mentioned he plans to use the AlphaFold to predict interactions between protein and molecules, and then construct a network, and eventually to build a whole cell. That probably will take 10 years. So actually, we also working on something. We're going to write something that in December, there's a cell paper published by Steve Quick and Aviv Regev and many others, and Emma Ludenberg, colleagues from our community. This is a very, very nice paper. Basically, they wrote everything that we want to say. So we think this is a very, very good idea. And from that on, we call this virtual cell. I think virtual cell is better name than a digital cell. But how to construct and grow AI cell, then it's still unclear even after reading this cell paper. So recently we have another article, we rephrase it, and we will publish very soon how to grow AI virtual cell. So we propose we have 3 data pillars and a closed loop learning. So the first pillar, like a grow virtual cells, we need to apply knowledge like literature, databases, multiomics from other tissue types. So behind the logic is the protein interaction in different species. So the second pillar is the static architecture for a particular cell type, like yeast. We should acquire images at a nanoscale eventually, and profile all the molecules, omics and phenotypes for the cell of interest. But more importantly is pillar 3, which is dynamic states. We need to generate different states by introducing perturbations and then measure the proteome before and after. So then we proposed using robotics to gradually realize closed loop active learning, because there are numerous perturbations. So we perturb using robotics and then generate a model, and then feedback generated, propose new perturbations, and then do this cycle and let the virtual cell to grow. So then we have a more concrete plan. Due to time limit, I'm not going to explain a lot of details. And we are actually discussing a lot with colleagues from the East biology and also computer science. So now we are we have a quite concrete plan. I also want to get your feedback. We want to generate data version 0.1 and then 0.2, and eventually when we finish, maybe in about 12 months, we want to generate the data V 1.0. So this is the current plan. Then there's a version 1, version 2, version 3 based on 3 pillars. I think I want to leave some time for discussion, so I'm not going to discuss a lot of details. So special thanks to Bogdan, Gil, Uwe, Jenny, Walt, Eric for supporting this AI proteomics initiative. I also want to use this initiative like AIVC to track collaboration from biology, from genetics, from genomics, from structural biology, imaging. So currently, quite a lot of people interested in it. Okay, thank you. I think time is running fast. I look forward to your question, comments.
Thank you very much, Tinan. It's a great talk with a lot of information and a lot of plans, and I'm happy that this initiative kind of started. And it's going to be very interesting, as you said, for many areas of the research. In my opinion, I think it will be would be an interest for all of our initiatives and the biology of disease at the HUPO. And I do believe that hopefully we will have a lot of type of collaboration together between our initiative and essentially your data. I do not see any questions in the comments as per se. Please, everyone who is in the current chat, use the chat to ask any questions if you want to. And until people try to put any questions there, I do have my general comment in many cases, of course, but I do not want to have full time. And I would like to open discussion for everyone as well. But my question would be, I think you are absolutely correct about the unpublished amount of data that currently all the labs have. And since I do believe that this talk is recorded and will be shared later with many much bigger audience, I would like to kind of echo Tinan request for the unpublished data to be used and loaded to this initiative. I think it's a great time that we can offload a lot of data that is not really being used for any publication. And there are many reasons why you're not publishing data. Maybe the biology didn't work. Maybe experiment wasn't kind of provide you results that you would like. But the data, mass spec data, is still there. And the mass spec data is still being identified, meaning the identification for the proteins are still present. Like for the purpose of what Tinan would like to use those, there's still a good data. That's why I think I would like to support this initiative, and I would like to ask everyone to look into these possibilities to share data that it's not going to be published by one or another reason, but still can be a great support for this initiative. I do believe further down we can actually provide more information, and I would like to make sure that HUPO support and put on the website area where the data could be uploaded, that it will be a common knowledge where and how to submit such data. And then clearly a lot of initiative that is currently building up that you, Tinan, described is really great example where AI could help us to understand biological areas of cell, the yeast cell and protein perturbation, etc. My question to you, and since you already kind of highlight a little bit of the plans for the next year, I do understand that it will be submitted for the EI part of this story. But on the, say, yeast initiative, how do you see when since I know a lot of yeast data already been kind of available before that, and when you will just set up this model, so to speak, computer model of the cell, AI based yeast, how that initiative would try to be shared between other users or other things. Clearly you will have a published papers, but I just tried to see what is your vision, how other, say, yeast biologists could contribute to your knowledge, maybe guide you where the biggest yeast-based biological questions could be answered based on this initiative.
Yeah, this is a very good question. So when we try to select the model for building an AI virtual cell, we consider several options. So eventually we decide the yeast. One reason is the yeast has a very small, relatively small number of genome. And another reason is that all the data for the yeast has been shared by the community. Yeast has a very, very mature community running for many decades. And all this data is shared, and they are not human sample. So we have no concerns. They will be, not the ethics issue. So when I talk to people from US, from Europe, from Canada, from other countries, so nobody has problem to ship yeast samples. Nobody has problem to share yeast data. So we plan to have a yeast consortium, AIVC yeast consortium, and my lab will generate a lot of yeast perturbation data, and we will share to the community, like what a CP Tech is doing. So maybe even easier than CP Tech, because CP Tech deals with human sample. So yeast sample can be, data can be shared. And yeast has also Matthias Mann and Max Raza, they publish a lot of high profile yeast data in Nature Cell journals. So these are very, very mature field. So, and from the other angle, I have talking to scientists doing super resolution imaging and cryo-EM, cryo-ET, those are structural biologists. So they also interested to work on yeast, and then they can share the data. So this also very easily to be shared. Another reason is I work with, talk to people working on robotics. So ideally we want to develop 100% closed loop active learning system, but to culture human cancer cell will be relatively costly and they worry about contamination. So we also worry about the contamination, but much easier compared to human cancer cell. It also grows very fast. So this AIVC East consortium is not fully functioning, but my thought is we should share all the data. And this is a demo for connecting the proteomic community and also connecting other communities. So in June and October, I plan to organize two in-person meeting in Wesley University to discuss more about details.
Okay, yeah, great, thank you. As I said, unfortunately, we don't have any question in the chat yet. We have another 2 minutes. The quick question from me would be, again, sorry to asking all the questions today, but it's our first initiative. Hopefully our audience will grow with time. How do you see synthetic biology will play a role in this initiative? Where you could see the basically a future fusion with the capabilities of the computer's power, and as well as what the biologists can now do with artificial amino acid, and how it will be incorporated into this project.
Yeah, this is very important. I forgot to mention this. Thanks for reminding me. Actually, one outcome, of major outcome, and one passion why people are excited about yeast is impact in synthetic biology. So another impact is drug discovery. So both community, I have not yet connect to approach synthetic biology community, but when we sort out how we organize as a proteomic community, I think I will approach them. So if you have people from the synthetic biology, yeah, I'm very keen to talking to them.
Glad to hear. Yes, definitely we should just make sure that the synthetic biology community will join this effort, because clearly it will be the two-way road, because they could provide to us organisms that are quite unique for analysis, and vice versa. We could provide to them data that they will not be able to achieve by any other type of technologies, as a genomic or RNA sequencing. Okay, Tian, thank you very much for your talk today and presentation of great initiative. As I mentioned, I think this is one of the key initiatives to have. in the nearest future, because all of our biological initiatives separately would basically, in my opinion, benefit from the possibilities and data that we can analyze together and try to understand the complex biology of disease. Thank you, Tinan. Our next speaker today, it is my pleasure to present Yu Gao. He is a professor of Beijing Normal University in China. And today he will speak about the new initiative under the biology of disease. That's a urine proteome initiative. Again, in my opinion, that's a quite important initiative to start. I think in many years we'll understand and know that a lot of effort was put into the analysis of blood as a main kind of biological liquid that we all been analyzed in our labs at some point. But I think much more possible areas to grow. And one of the area that I could see a urine proteome initiative would be a great part of this story. And again, Yuhe, thank you very much for joining us today to represent this new initiative on the BDHPP. And stage is yours. Thank you.
Thank you. Thank you for giving me the chance to introduce the Urine Proteome Initiative. Let me try to share my screen. Why do we have to do the Urine Proteome Initiative? There are a few reasons. Actually, I'm from the medicine background. The most important thing in medicine is early diagnosis. Without early diagnosis, even with drug, we don't know who to give and when to give to the patient. We don't even know where the patient is. So the most important thing in medicine is early diagnosis. If it's too late, we don't give the doctor a chance to save us, to help us. So let's focus on early. But when we talk about early biomarkers, we always think about the closest place to those organs, sick organs. So when we think about brain disease, we thought of CSF. Of course, people would like to concentrate on blood because blood has uh, material exchange and energy exchange with all the organs. It connects directly to all the organs. That's why we used to work on blood. That's reasonable. But the problem is when we think about the homeostatic mechanisms, the homeostatic the internal environment, homeostatic of internal environment theory, and we thought about maybe the earliest change may not be in blood because when the homeostatic mechanism is fully working, the changes are removed from the blood. They're not there. It's so hard to find disease-related change in blood, especially in early stage. It's so hard to find it stable. It is removed constantly. The change is removed constantly from the blood. Where do they go? One of the main place is in urine. So actually, blood is relatively stable at early stage, but urine accumulates all the very early changes associated with the disease. Then, with the theory, uh, for many years, a lot of studies have been done on animal models. People rarely do animal model study in the urine field because urine is so easily collected from humans, and it's not that easy collect from animals. It's so easy to collect a lot of samples from human being and with completely invasive. Uh, so a lot of study have been done on human samples, but not that conclusive. I think the reason why is because there's so many confounding factors when you study with human samples. Some people smoke, some people drink, some people drink tea, some people drink different coffee, and they all have impact on the urine protein too. So that's why it's so hard to get uh, relatively impeccable conclusion that the changes that we see in the urine is the is the reflection of the pathophysiology of the body. So the good way at the early stage, the good way is to use animal models. Use animal model, we don't have any confounding factors, almost completely removed. The only difference, they eat the same thing, they drink the same thing, they stay in the same place. The only difference between the two groups is one is is diseased group, one is control, healthy control. So another advantage of using animal model is we know when the disease starts. For human being, we never find a person who was diagnosed with cancer yesterday was actually the cancer started to grow yesterday. The cancer started to grow long time ago. We only diagnosed yesterday. But in the animal model, if we inject the cancer cell yesterday, then that's the starting point of the cancer. So it's the as early as possible as we can get in animal model. We can't do that early uh, study in human being at all. With the advantage, we can see that in urine protein, a lot of disease can be reflected at very early stage. Even very subtle changes of the body can be seen in urine protein. There's so many evidence. I don't have time to talk about. If you are interested, uh, we will do some more talk. And actually, at the same time, more and more evidence have been collected, have been shown from the human studies too. Those the changes in urine protein may not uh, just for fun to see in animal models. It's really usable for human disease early diagnosis. There's also a lot of examples too. Uh, Actually, just as Tiannan talked about the AI, it will change the whole world. The AI, it's revolutionary, it's great, but the quality and the quantity of the data. I think the best quality data is about human health. And there's so many things about human health. You can see, you can use fingerprints, you can use the retina, all kinds of things to represent human being. But actually, uh, urine is the most dynamic. It reflects everything in your life. You drink a cup of tea, it reflected in urine. You have a meal, a big meal, small meal, they're all reflected in urine. So urine is probably the most comprehensive dynamic data that we can have for human health. So I think that's the most valuable things that we can have. With thousands of proteins in urine and thousands of metabolic small molecules from urine, we can have a very clear picture of the human health. I think with those data, with the help of AI to analyze, eventually urine will change the medicine very significantly. We will push the medicine to a very early stage, much earlier stage than it is now. Actually, we published it, the whole theory a long time ago, but it's uh, never been noticed. It seems never been noticed to me. I think it's has a big potential for the biologists and all the researchers and doctors, and companies, and governments. Actually, Tenman did the presentation for me to apply to start the urine protein initiative. And so far, we have myself and Tanya from UK and Anna from USA and Tadashi from Japan as chair and co-chairs. At least we have a group now. Actually, we have started the academic exchange long time ago. As soon as we started the, the theory that urine may be a better biomarker source than blood, earlier and more sensitive. And we start with discussion groups because we don't have any official medias. So now based on WeChat, each WeChat group, we have maximum only 500 people allowed in each group. Now we have over 3,000, many different groups, and we share the up-to-date urine-related research almost every day in those discussion groups. And we also share short videos and other materials related on the WeChat public account. We also had 3 Urimarker conferences before. Those are like the only meeting with the only theme, which is urine biomarker. Recently, we proposed that we should have a urine project, a world urine project, because I don't think it's really being noticed with such a good potential for the benefit of human health. Thanks Tanya and Bogdan, who co-proposed the World Urine Project. We published that short piece of paper last year. We hope that this field can be recognized and we can collaborate for the whole, all the scientists from over the world to have some standard, to share database, to promote translation, overall to benefit the human health, to make the early diagnosis possible. Actually, we make a little more progress in China Urine Project. We had the first meeting last September, and we received some support from over 100 professors and higher level managers from companies, hospitals, and academic institutes. And we published a proposal in the one of the almost official scientific publication, publishing house, Science China Life Science. And we're going to have a second meeting next year, next month, talking about a little more details. So far, we have received a good response from many different companies and many different biobanks and hospitals. And the idea is, so far, we don't have any funding yet. So we wanted to use the spare resource to just get started. Some of the biobanks, they have urine samples. They do not use it, so it's a waste. And some of the companies, they have spare machine times. If you don't use it, it's lost too. So we wanted to combine those resources and put together to generate data which will be will benefit the early diagnosis of human beings. At the first phase, we hope to have some healthy human urine proteomes. Personally, I tried very hard to promote this field. Those are all the PPTs I gave, talks I gave in the last few years, over 100 talks. I go out almost every 2 weeks to give talks to promote the field. And one important thing I would like to show is there's a big problem. People don't save urine because it is too expensive. Urine is too dilute, and you use a lot of space uh, in the refrigerator, in the freezer, to save urine. So we developed a method, which I call it UriMan. Basically, it's just filter the urine, remove the water part of the urine, and use the membrane, which is either nitrocellulose or PVDF, or whatever membrane that is sticky to proteins. So the protein will stick on the membrane. And when the water is removed, the, it becomes a piece of paper. And when you put that piece of paper in under the dryer or hair dryer, then it becomes a dry piece of paper, and seal it in a vacuum bag with no water, no oxygen, no air, nothing, and then it it will last forever almost. You don't even have to put it in the freezer. You can leave it at room temperature, put it on the bookshelf, or with the medical history of the donor. The good things with this is it's so easy and so simple and so economical. We don't even have to wait until we are sick to collect urine. We can do it regularly and save those urine samples. Eventually, everybody will get sick. And then we can find samples collected even before we get sick. In that case, we can push the diagnosis to an earlier stage. So I try to promote these things almost everywhere. I hope the government will see the benefit of doing this, and we save a lot of sample uh, even when we are still healthy. And so when we get sick, we can come back to analyze those samples and make early diagnosis for the future for the people. And I think we should be more visible. And I went to the HUPO meeting in Germany, but we don't even have a place to talk. We don't have a room for people who work in the urine to talk, and we don't have any chance to give any oral presentation. I think we should be more visible since it's so important, so uh, big potential for the health of human being. And so far, because of those current political problems, I think with this initiative, the scientists from over the world can have a good scientific communication and cooperation for a good purpose. And I think it's good for HUPO too. And there's so many uh, different fields that we can work on in HUPO, but urine is one of the closest to translation, to the diagnosis of human disease. So I think the potential is high. Uh, that's that's my talk. Thank you so much, Ms. Bogdan, for giving me the chance to talk. And I would like to save some time so we can have some communicate, answer some questions.
Okay. Thank you very much, Yohe. It was very interesting and important talk from you. Thank you. I definitely think that it would be much more coming, and I hope that this talk today and further HUPO initiative would have more and more attention paid to the urine proteome. Before I ask kind of my question, we do have a question in the chat from the Christopher Sutton, and The question is about, is the peptides included in this PDX membrane that is used? And I think it's important question to share here.
Good questions. And I don't think all the peptide, all the proteins, all the small molecules are that sticky to nitrocellulose or to particular PTMs. But even without those, there's still most of the proteins. When we wrote the paper, I referred the data from the manufacturer, the nitrocellulose manufacturer. They say almost all the proteins are there. But with those proteins, it's good enough for me, for us. We have tried many different diagnoses, or at least to find very easily to find differential proteins for almost any disease in animal models, almost any conditions like fibrosis, cancer, and metabolic diseases, neurodegenerative disease, and autoimmune cancer. Uh, Once we start looking at urine, you see so many things are changing all the time, and there's some consensus at particular time point for particular conditions. And I think the advantage of urine is it's a combination of many different factors. But with AI, all those factors will be distinguished, will be identified. Uh, the, Eventually, we'll see which biomarker is closely related to this disease, which one is related to green tea, which one is related to beer. And it's not that difficult with the help of AI and the computing power of current era. I think it would be okay. Good.
Yeah, thank you, Yuhe. And I'm actually glad that Tinan is part of this conversation. And that's exactly
Yeah.
why I think, the
Yeah.
initiative that Tinan is leading is important, because all the data
Yes.
that we
Definitely.
could produce could come to the AI group for such analysis. And clearly, I would like them to have an extra initiative that will be urine related. I think we do
Great.
have a question about the peptides kind of discussed, and I think it's important one. And I do believe that it's another question that I would like to raise, that always when we speak about the urine proteome, I think it's a very specific, you don't need to convince me. I do have my own evidence for that. And I think the two other important part which we would like to note here in this talk, in my opinion, that the upper part of the proteome, which is basically what kind of proteins could go through the kidney to the urine. And usually, if you're talking to the generally MD, and that's about 55 kilodalton is count to be around the natural filtering system of what the kidney provide as to the proteome of the urine, unless kidney has a problem. And that's why I think the importance of the kidney health should be raised here as well as, in my opinion, the advantage of why urine proteome is so important, because urine proteome could go towards the home test, which is essentially not possible for the blood in many cases. That's two subject I want you to have a quick answer.
Okay. Actually, I would like to talk about that proteins in urine of healthy people. And we used to think only small peptides, small proteins are in the urine. But actually, we have thousands of proteins, even with healthy people, and some of the proteins are not even that small. Let me make an example, like albumin. Albumin is not very small. There's a lot of albumins in blood, but there are also a lot of albumins in urine too. Actually, we did a small study. Why, how, and kidney knows what albumin it's old albumin. It should be removed, should be thrown into the urine. I tried very hard to find the difference between the albumin in blood and albumin in urine. I didn't find any particular site which is unique for blood or in urine. I didn't find any particular unique uh, post-translational modifications in blood or in urine. But recently we did publish a paper. We found out that the albumin in urine has higher possibility of forming the disulfide bond. And the albumin in urine, I hope I didn't say it wrong. There's more disulfide bond for the albumin in blood, less disulfide bond for the albumin in urine. That's why the albumin in urine is probably less functional. That's why they are uh, maybe more, instead of like a big three-dimensional structure, is maybe like a thread. It's easier to filter from the kidney, and they're why that's probably why they're in urine. So we see a lot of post-translational differences between proteins in blood and urine. That's probably another mechanism. It's probably not only the size. Uh, there's big proteins too.
Yeah, great. Thank you for this answer.
Oh, what about the second question?
The second question about importance of the urine as a possible in-home testing
Oh,
compared to
yes, yes,
blood.
yes, 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 yes. It's more likely to be used at home or in other place. It's completely invasive. It's that's a good, good, good.
We have one person, Alessio, from the chat. He is a hand. Please, Alessio, if you could join us for the asking question or comment, please, you're welcome to do so. You muted.
But we can always discuss later, even after the meeting.
Alessio, can you ask your question?
Hello, can you hear me?
Yes.
Hi, yeah, please.
Okay. So I have a question about post-translational modification in urine protein, like uromodulin. Have you found maybe some modification in uromodulin related
Uh,
to?
there should be. I don't remember the details. Since we work on proteomes, it's old, old, always thousands of proteins. Uromodelin is such a high abundant proteins. It's shown in almost all the conditions. And it's very easy to change, and specificity may not be that great, but it changes all the time, as I remember. I don't know exactly the details, but if you have any particular question on particular disease animal models, and I can check the data for you.
Thank you very much.
Sure.
Okay, thank you for your presentation.
Thank you for
I would
giving me
say
the
that
time.
this is very important initiative to join. I strongly suggest to people who is interested to join the WeChat that you have showed before, and be part of the larger community of the scientists who is actually interested in this initiative, and maybe sharing data or actually asking question and discussing the urine proteome. That's a big field. And I do believe that all this data should actually essentially go to the EI initiative as well, to look from this large data set and try to see what kind of code of disease we could see earlier in the urine And clearly, it's non-invasive type of samples. That's why we all would like to use this and try to see if this would be the way to get earlier diseases detection. Again, I would like to conclude this meeting today. And I'm very pleased to see that we have a very great two initiative being presented today, AI and urine proteome. Those are very important initiatives under the biology of disease in the HUPO. And I would like to make sure that we continue this, and the next few initiatives will be presented in June. And you all welcome to join in on our next meeting. The HUPO will officially show the dates for the next presentation. And again, thank you very much, Tina, and thank you very much, you here, for your presentations today. And
Thank
I'm
you
looking
for you
forward to
organize the
working
meeting
with.
for us. Thank you.
Thank you.
Thanks. Bye,
Nice
everyone.
day.